This rendering represents the culmination of a rigorous biological gauntlet. Two highly specialized gametes must navigate a specific sequence of chemical and mechanical events. We begin inside the female reproductive tract, where the environment is actively preparing for the release of the first gamete. This diagram outlines the ovary before ovulation. In panel A, the mature vesicular follicle bulges at the surface of the ovarian stroma. An abrupt surge in luteinizing hormone, or LH, triggers the primary oocyte to complete its first meiotic division. This LH peak increases collagenase activity, digesting the follicle wall. Muscular contractions then physically extrude the newly formed secondary oocyte, as seen in panel B. It immediately begins its second meiotic division, halting in metaphase until fertilized. Ovulation is a precise hormonal synchronization that ensures the female gamete is biologically mature and properly staged before it enters the uterine tube. Once expelled, the secondary oocyte is suspended in escaping follicular fluid, shielded by a series of distinct protective layers. Positioned alongside it is the first polar body, a cellular byproduct of the first meiotic division. The innermost protective barrier is the zona pellucida. This is a thick glycoprotein shell that completely encases the oocyte. Surrounding the zona is the corona radiata, which consists of several layers of radially arranged granulosa cells. During ovulation, the finger-like fimbriae of the uterine tube move over the ovary, capturing the oocyte cumulus complex. Fluid currents created by ciliated mucosal cells and the rhythmic peristalsis of the tube wall push the oocyte deeper into the reproductive tract. Its destination is the ampulla. This is the widest section of the uterine tube and the typical site of fertilization. The timing is critical. Once released, the unfertilized secondary oocyte remains viable for approximately 24 hours. The reproductive tract has now positioned a fortified, mature gamete in the ampulla, establishing the window for the arrival of the male gamete. While the oocyte waits in the ampulla, the male gametes begin their own parallel journey through the reproductive tract. This chart maps the population attrition. The journey begins with the deposit of approximately 200 to 300 million spermatozoa. Most of this transport is actually driven by muscular contractions of the uterus and uterine tube, rather than swimming motion. Only 1% of the sperm manage to enter the cervix. Millions more are lost navigating the uterus. By the time the survivors reach the ampulla, only 300 to 500 spermatozoa remain from the original hundreds of millions. However, physical arrival is not enough. These sperm are not yet capable of fertilizing the oocyte. While the migration serves as a rigorous endurance filter, the surviving sperm must still undergo a series of molecular changes to breach the oocyte's barriers. To acquire this capability, the sperm must undergo capacitation. In humans, this period of biological conditioning lasts approximately 7 hours. Observing a single spermatozoon interacting with the uterine tube reveals the specific molecular shifts required. Interactions with the mucosal epithelial surface of the tube trigger a specific sequence of membrane alterations. During this process, a glycoprotein coat is removed from the plasma membrane overlying the acrosomal region of the sperm head. Simultaneously, seminal plasma proteins are stripped from the surface of the acrosome. These changes increase membrane permeability, facilitating essential ion exchanges like the vital influx of calcium. The physical result of these structural alterations is hyperactivated motility, which is characterized by significantly more vigorous tail movements. Only after completing capacitation is a sperm fully capable of passing through the corona radiata cells. The female tract has effectively transformed the sperm from a transport vehicle into a biochemically active cell, capable of navigating the final barriers. This scanning electron micrograph shows the result of that transformation. A few hundred capacitated sperm now swarm the surface of the oocyte in the ampulla. Phase 1 is the penetration of the corona radiata. Sperm navigate this layer using hyperactivated motility and hyaluronase to move between follicular cells. Phase 2 begins when the sperm's head makes direct contact with the glycoprotein shell of the zona pellucida. As shown in panel B, the sperm binds to a specific zona protein known as the ZP3 ligand. This interaction is highly species-specific. The ZP3 ligand ensures that only sperm from the same species can initiate the final stages of penetration. Phase 1 tests mechanical and enzymatic navigation. Phase 2 provides the final chemical verification to proceed. Binding to the ZP3 ligand initiates the next critical event, the irreversible acrosome reaction. This reaction involves multiple point fusions between the outer acrosomal membrane and the plasma membrane of the sperm head. These fusions create a series of perforations across the cap of the sperm. Stage 3 illustrates the resulting payload. Enzymes like acrosin and trypsin-like substances pour out. The enzyme acrosin chemically digests a localized, distinct physical tunnel through the zona pellucida. The sperm head moves through this path, driven by its hyperactivated tail. This movement continues until the sperm's plasma membrane physically touches the oocyte's plasma membrane. The final barrier has been breached.